Yeah, so good afternoon and good morning, everyone. I am Radhika Bansal from research scholar from Jaypee Institute of Information Technology, along with my mentor, Professor Pami Goba, Head Department of Biotechnology. Would like to thank conveners of PSA 2022 for organizing such a wonderful conference and giving us the opportunity to present on investigation of accumulated heavy metals in the leafy drugs commonly used in Ayurvedic formulations collected from various ecological zones of India. We firmly believe that this is a very relevant topic as Ayurveda is gaining importance globally. And being natural in origin, herbs are considered to be safe and without any side effects. Ayurveda, the concept of Ayurveda in India has developed back, believed to develop back in 2500 and 500 BC, and it may be older also. And being the important part of medical system, it is also called a science of life. So the raw herbs are sold in the open market over the counter and are prescribed by the traditional practitioners for the ailments such as diabetes, hypertension, um, uh, disorders related to various vital organs such as liver, kidney, etc. Um, so Ayurveda for self-treatment is preferred over any other medicinal system in the developing countries, where about 70 to 80% of the population rely on the herbal medication. And even the modern medical system relies on the phytochemicals derived from the herbs, such as colchicine from Colchicum autumnale, Soralia, Soralin, which is commonly used in treating various skin disorders derived from Soralia corifolia, etc., are used for the treatment of various diseases. So while therapy is being widely used due to its potency and less side effects, increasing heavy metal contamination in the soil due to anthropogenic perturbations and use of chemical-based fertilizers and pesticides, wastewater irrigation, etc., is posing serious concern due to the accumulation of heavy metals in the soil, which can be uptaken by the medicinal plants and can get accumulated in the human body upon consumption, which causes toxicity to the various vital organs, formation of DNA adducts, protein crosslinks, and causing point mutation and genotoxicity. So the mechanism of different heavy metals which we have studied includes chromium, mercury, lead, arsenic, and cadmium. So when the hexavalent chromium is uptaken by the cell, inside the cytosol, it gets converted into its intermediates and reactive oxygen species are generated, which upon entering the nucleus causes chromium DNA adducts and protein crosslinks, generating point mutation and causing genotoxicity and cytogenetic effects. Mercury, on the other side, might be epigenetic carcinogen, and it interferes with the gap junction intercellular communication, and which is linked to the upstream alteration of intercellular redox tone caused by the inhibition of antioxidant enzymes containing selenocysteines. Lead toxicity is linked to the inhibition of heme synthesis because it inhibits the enzymes that catalyze the hemoglobin synthesis. Cadmium, on the other hand, is related to the neuronal cell damage. So, and arsenic toxicity, it causes DNA methylation. And DNA methylation, hypo-DNA methylation can potentially lead to aberrant gene expression and oncogenic growth in the liver. Therefore, the present work plan includes the sampling of leaf samples, Calotropis procera, Cinnamomum tamala, Phalanthus paternus, Ricinus communis, and Cannabis sativa from east, west, north, and south zones of India, and the authentication of these herbs from the expert botanist and heavy metal analysis in them. So the leaf samples of these commonly used leafy herbs were sampled from different ecological zones of India and were authenticated from the CCRAS labs of Ministry of Ayush, Government of India under the supervision of expert botanists. The heavy metal contamination or concentration in these herbs of chromium, arsenic, cadmium, lead, and mercury were studied. So where we can see that chromium contamination concentration in Calotropis procera, Phyllanthus fraternus, and Cinnamomum tamala exceeded the permissible limit, which is 2 ppm, prescribed by Ayurvedic Pharmacopoeia of India and World Health Organization. And also we can see that 1.6 to 1.79, these are very nearing to 2 ppm. And if the agricultural practices are not controlled, they may increase in near future. On the other hand, we can see for mercury, the permissible limit prescribed by WHO is 1 ppm. And in cannabis sativa in the east zone, the mercury concentration is 14.31 ppm, which is extremely high. So the green bar depicts the permissible limit for chromium, and the red bar depicts the concentration above the permissible limit in the three herbs. And also Cinnamomum tamala, where the chromium concentration is so high, we can see that it is the commonly used spice in every Indian kitchen. Although arsenic, lead, and cadmium concentration are within the permissible limit, but some content of these heavy metals was also found. Whereas for the mercury, we can see the concentration of mercury is extremely high in cannabis sativa in the east zone. Therefore, we say that prevention is better than action. So phytoremediation proves to be a very effective measure to remediate the heavy metals from the soil, which includes phytodegradation, phytovolatilization, phytoextraction, phytostabilization, and phytofiltration of these heavy metals. Plants tend to accumulate and uptake these heavy metals in the roots and translocate them in the shoot, thereby rendering soil free from heavy metal contamination. So to study this, we have worked on the phytoremediation of hexavalent chromium by using two plants, Cicer aurantium and Vignamungo. And we have studied the toxicity of hexavalent chromium in these plants and the remediation potential of both these plants, including percentage of remediation, which is the extremely important aspect for a plant to be a good phytoremediator, and also translocation and bioaccumulation factors. So displayed are the reference pictures for how the experiment was conducted, starting from the germination of the seeds, sowing them, and the growth over the period of 3 weeks. So as we can see that percentage remediation of 72.17% was achieved by Cicer aurantium and 76.67% by Vignamungo at 400 ppm of hexavalent chromium contamination in the soil, which is a very good uh, percentage, we can say. Therefore, we may conclude from this that concentration of the selected heavy metals in most of the herbs are within the permissible limit, but chromium contamination and mercury content exceeded. Also in the west zone, none of the sample had heavy metals above the permissible limit. So it is recommended that for the preparation of any decoction or concoction or even consuming the herbs in the raw form, west zone is the best zone to sample the herbs from. And also, heavy metal contamination can be controlled by applying good agricultural practices and obviously phytoremediation, which is a very effective measure to remediate heavy metals from the soil. These are the references. And in our future prospects, we are trying to use different types of plant species for the remediation of chromium, mercury, arsenic, cadmium, and lead by using them as potential phytoremediators. And also we are trying to isolate microbes that can sustain a high concentration of these heavy metals, and also which can convert the heavy metals into their intermediate forms, which are not readily uptaken by the plants, so that we may increase the remediation of these heavy metals in synergy with the plants. Um, so at last, I would like to thank Jaypee Institute of Information Technology, Noida, India, for providing us the extremely good facilities, lab facilities, and of course, Ministry of Ayush, Government of India for funding the present research. Many thanks to our research group. We all are working on microbial and phytoremediation of inorganic and organic pollutants. under the supervision of Professor Pammi Goba. Ma'am is constantly supporting us on every step. So thank you, everyone, and for your patient listening. And I wish you better tomorrow, healthier tomorrow with Ayurveda. Thanks a lot.